Good day. This is the Professor, CBMD PhD, and today's Wednesday, May 31st, 2023. It is 4:13 and 16 seconds, a.m. Eastern Daylight Time, and today's research, bladder cancer. So let's hop to it here. Researchers identify immune cell that help kill bladder cancer tumors. Study also identifies potential ways to make immunotherapy more effective against this deadly cancer. Date September 12, 2022. Source: Mount Sinai School of Medicine. Researchers have made two important discoveries about the mechanism by which bladder cancer cells foil attacks from the immune system. The research could lead to a new therapeutic option for patients with these types of tumors. Advanced bladder cancer is aggressive, and patients generally have poor prognosis. Several immune checkpoint inhibitors have been approved by the Food and Drug Administration for bladder cancer, but they only sustain good responses in about 20% of patients. When people get cancer, a type of immune cell called a natural destroyer cell swings into action to try to kill off the tumor cells. However, the tumor cells are often able to foil the attacks from the natural destroyer cells. Researchers have found a subset of CD8 T cells that adapts to tumor evasion strategies by appropriating innate-like properties traditionally ascribed to natural destroyer cells, offering a strategy to reduce the tumor cells' ability to fight them off. And to create additional destroyer cells, the researchers showed that they could induce CD8 T cells to express a molecule known as NKG2A on their surface, allowing them to behave more like natural destroyer cells. And this study showed that NKG2A is associated with improved survival and the responsiveness to a cancer-fighting immunotherapy known as PD-L1 checkpoint blockade. The second discovery concerns cancer cells' ability to resist PD-L1 checkpoint blockade therapy. The researchers noted that tumors can maintain expression of a protein called HLA-E on their surface that can resist the T cells because the HLA-E binds to the NKG2A molecule that disables the T cells' ability to fight. An immunotherapy that specifically targets the HLA-E NKG2A axis could be an effective way to attack cancers in these patients. And these findings suggest that antibiotics that block both NKG2A and PD-L1 could be a more effective treatment strategy for patients whose bladder cancer tumors have both high levels of HLA-E and NKG2A positive CD8 T cells. And these findings provide a framework for future clinical trials that combine a therapy that blocks NKG2A with other immunotherapies in these tumors. Immune checkpoint blockade is a leading type of cancer immunotherapy that targets the PD-1, PD-L1 pathway in order to re-engage exhausted CD8 T cells in the fight against tumors. Research has shown that responses to PD-L1 blockade in patients with bladder cancer are influenced by an additional immune checkpoint axis identified recently in other cancers, NKG2A, HLA-E. Researchers profiled tumors and blood of bladder cancer patients across various stages of the disease and studied all specimens immediately after surgical removal from patients to ensure they could capture live immune cells and examine their function. The study integrated several cutting-edge single-cell technologies and leveraged publicly available datasets to the Cancer Genome Atlas and the IMvigor 210 trial of atezolizumab, an anti-PD-L1 immunotherapy. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and Leader of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have an awesome day.